Welcome everyone. As usual, we'll get started around 5 after the hour. Let everyone settle in. So if you're rushing around, you want to go get yourself something to drink or get comfortable, it's a good time to do it. Welcome everyone. Just so you know, we'll get started at 5 after the hour. We put it in the chat. But if you need anything or you're having any type of technical issues, just shoot us a chat and we'll try to address it. Shouldn't be having any issues. And we'll get started in about, I think we're down to a minute or two. Yeah, 1 minute. Folks are still joining. All right, welcome everybody. We have an exciting webinar today. We're with Elucidata, and they are going to talk to you about their Liver OmixAtlas that they've spent some serious time creating. It's probably the world's most comprehensive and curated liver data collection out there. And let's get started. So, rules of engagement. If you have questions, let's ask them all. You can ask them during the talk, but they won't get addressed until the end, until the Q&A session. So put them in the chat or the Q&A box, and we will be addressing those. We'll moderate those with the panelists. Sometimes you're going to have really good questions. We may have some good questions, etc. You can ask questions if they're all appropriate, they'll all get answered. And then everyone will have access to this recording, and it will go on demand afterwards if you want to go back and rewatch anything. So we're going to do speaker introductions, a brief industry perspective, and then Elucidata presentation and demo. And then after that, the Q&A. So I'd like to introduce to you Abhishek Jha. He's the co-founder and CEO of Elucidata, and Shefali Lathwal. She is the Omics and Drug Discovery Scientist Lead for Elucidata, and they both will be talking and presenting today. My name's John Conway. I'm the founder and Chief Visioneer Officer for 2015 Visioneers. And I'm going to give you a little industry perspective and background before we get started. So just talk about some minor definitions and some scientific informatics and guidance. We're working in this space, and we have the pleasure and luxury of working with great companies like Elucidata. So FAIR principles in model quality data. So FAIR means findable, accessible, interoperable, and reusable. The reason this has evolved to this point is because scientific discovery is complex, and a lot of times it gets underestimated, even by some of the best in the world at what they do. And we've evolved to this point where a lot of this data today that we have isn't fair. And is missing a lot of contextualization. It's missing really the culture of data as an asset. Data is a high value entity in our lives, especially because we've now evolved to the point where we have machine learning, artificial intelligence, predictive methods that rely on this data, or a type of fuel, a high quality fuel is needed to run these machines. And it's not in a state where we're getting the most value out of it. And so we need to change this. And the reason this is really critical now is because multi-omics is going to really drive innovation and discovery moving forward, and only if we have fair and model quality data. So model quality data is high data integrity, highly contextualized, assessed with confidence, and then the applicability has been established. In other words, it's data that you're going to, without question, be able to use in your predictive models. And that's going to allow you to become an in silico or model first type organization, which many organizations are starting to get there on. And when you get to that level, you're talking about a 40% efficiency gain in some of your areas. It's amazing. And I've experienced that, our team has experienced that. An important part, in my opinion, is scientific data management and informatics practices and capabilities are in their infancies. There are a plethora of things that we can do and that we are doing to boost scientific data. We're putting different dimensions on it so that you get even more value out of it. So, but like the background picture here is, if you don't do this today in your multi-omics environment, you're going to have a circus on your hands, and it's going to be a very expensive circus. So buckle down and try to get this done. So both an excitement and a challenge is multi-omics, in my opinion. You have vast amounts of data, diverse data sets, deep analysis methods, pipelines and processes. And again, it has the ability to drive innovation and discovery like nothing we have seen in the past 30, 50 years of drug discovery. So one of the things that we've done in assessments for clients is to build out the whole landscape, the whole roadmap for a microbiome multi-omics environment. And you are going to need tools like what Elucidata provides, curated data sets, what they provide, but you're also going to have other tools, other processes in place there to make that enterprise environment work. And when you basically handle the 6 high-level processes: request, sample, test, experiment, analysis, and reporting, you will be able to make this environment work really well. And what you're going to see today is a perfect example of making model quality data, fair data, and for specific liver applicability, you're going to see what an amazing job that Elucidata has been able to do with that. So I'll hand it over to Shefali now, and she'll and Abhishek will take it from here.
Thank you, John. So I'm going to share my screen now, and first of all, Abhishek will give some remarks, and then I will take over and do a demonstration for you. Please let me know. Is my screen visible?
Yes. Yep.
Okay, wonderful. So Abhishek, you can take it.
Thanks, Shefali. And thanks, John. Thanks for having us over, and thanks everyone for attending this, taking time out of your days to join us today, this morning. We are very excited to announce and release Liver OmixAtlas. Shefali, my colleague, and I will walk you through this. In the first bit where I'll introduce the company and as to how we have approached this problem at large. So let me briefly walk you through the agenda of the next 50 minutes. Um, I'll give you a high-level introduction of what Omics Atlas is and how it originated and how it's a reflection of our approach to ML systems, specifically in life sciences and biotech industry at large. Then I'll hand it over to Shefali, my colleague. She will walk you through the Liver OmixAtlas, what ML readiness means for you as a scientist, be a biologist or a data scientist, a computational biologist. We're also going to talk about how you can take Liver OmixAtlas to where you are, to your own computational infrastructure. And then there will be a live demo which Shefali will run through. And at the end, again, John, me, and Shefali will come together to talk about, to do a small panel discussions. So we'll leave around 10-15 minutes at the end. Now, at a high level, this is what defines our approach. There's a lot of promise and excitement around ML systems at large in industry, right? You hear often about companies and groups talking about how they have an ML AI model which can predict the best patient segment, for example, that can predict the best target, or that can predict the most relevant small molecules that will affect that target. And there are a huge variety of such examples that we hear often. And at a high level, at a very abstract level, the ML system is made up of two parts, right? Model, which is the code or algorithm, and the data. And a bunch of progress that we have seen in the last 10 years in this industry and others has been driven mostly by model. So what we mean by that is, specifically as a data scientist, you download different versions of the model from Git or some paper that has just come out. And the data piece is more or less static, right? Like where you download a template of data that is predefined, pre-curated, and you test different versions of the model to see what would work for you. We believe that leaves a real enterprise level ML initiative within a company at risk, because the data is still an unknown, where you find the real life data and that does not quite fit, and you end up spending a lot of time just curating it, getting it ML ready, and that is often not resourced well, not thought of well, and that is a risk that we want to eliminate or minimize, so that your ML initiative within your company is successful. And we believe as a company that that is also a macroscopic challenge, if you will, right, which has plagued the industry, and is our worldview that has so far prevented us from unlocking the value of data that is being generated at an incredible pace. So you see what is on the orange curve is the Moore's law, where the growth in data has been exponential. And in blue, you see what is been named as Irum's law, which is Moore's law spelled backwards. And you get the idea, right? That bringing a drug to the market has never been more expensive and more, in fact, slower. And again, I'm preaching to the choir in this audience. We believe this has been discussed at length, podcast, books, and whatnot. Our viewpoint is that a lot of this data that is being generated is semi-structured to begin with. That's true for public data. That's true for proprietary data. And that's the problem that we have committed ourselves to address in the next, we have been working on this for the last few years, and we'll continue to focus and develop our technology around solving this problem. Shefali. And again, this is a slightly different look to the worldview that we have. On two axes, we have captured the availability of data, which is a spectrum. Something that is readily available, some things that are available but you're not quite aware of. Some things might require a small password that you have to fill in the web portal. And somewhere there are more detailed, complex applications that you have to fill an IRB of sorts. And in some cases, you have to do all of that and pay as well, right? So the availability alone is a spectrum. And then the other axis is ML readiness. We'll define that in more detail as we go along. And that's also a spectrum, right? Where some data sets, some repositories are more ML ready than others. For example, TCGA is on more on that right side of the spectrum than compared to, let's say, Geo. And what we have focused on is the second quadrant, repositories that are reasonably, easily available, but not quite ML ready. And our mandate so far has been to take this from the second quadrant to the, Shefali, can you go back? to the second, from the second quadrant to the first quadrant. And we have done this for 400,000 data sets over the last many months. And today we'll be focusing on a very small subset of that, which is what we're calling the Liver OmixAtlas. And that's a very small piece of what we have done, but it's a pretty impressive and to our knowledge, like a pretty comprehensive Omics Atlas, which includes around 7,000 data sets, 1.7 million samples from a huge variety of sources, dozen different type of data types across different organisms, diseases, and so on and so forth. Again, this is the overview. This is what we want to share with this community and get your feedback. With this, I'll stop and I'll hand it over to Shefali, who will walk us through the rest of the presentation and the demo.
Thank you, Abhishek. Abhishek just talked about how Liver OmixAtlas takes available data and makes it ML ready. So in the next 30 or so minutes, I'm going to show you what ML readiness means and what it can bring to your work. And the second piece of it is how you can take these ML ready data and work with them in your own computational infrastructure, wherever you are most comfortable working. So you can think of ML readiness as consisting of two major components. One is curated metadata, and the other is a consistent structure. So what curation of metadata means for you as a liver researcher is that it can help you discover data that are highly relevant for you, because we have tagged the data as well as the samples within those data with consistent standardized terms, irrespective of where they're coming from. So you saw earlier, these data coming from 10 different sources, and all of the metadata associated with them have been standardized. And I'm going to show you an example of this in just a few slides. The second part of ML readiness, which is consistency in structure, gives you the ability to integrate and combine data from multiple sources. For whether you're doing machine learning downstream or you're doing some other statistical analysis. And
Sorry,
we're also...
Can I interrupt you? Can you turn on your video?
Sure. Just a second.
Thanks.
Sorry, am I visible now?
Yes, you are.
Okay, wonderful. Thank you. Yes, so the second piece of this puzzle is consistency of structure. And what this allows you to do is really combine and work with data from very diverse sources together. And the third part, we're also going to talk about structure in some detail when we go to the demonstration. And the third part of this is we recognize that you may already have a computational infrastructure that you prefer for privacy or other reasons, something that you're already comfortable in. So these ML ready data also need to be available to you to work wherever and whatever environment you're using. And we'll also see that later in the demonstration. So if you are a liver researcher, you may already be working on several problems like the ones you see on your screen or similar ones. And you may be working on one or more such problems in your organization. And while these are very, very different questions that you may be trying to solve, what is common between them is that they're all very data heavy. So all of these problems require large volumes of data as well as very diverse types of data to solve. So for the rest of the session, I'm going to take one of these problems as an example. I'm going to choose predicting hepatocellular carcinoma patient survival, and we're going to look at ML readiness and how to use these data in context of this particular statement. So let's start with this example problem where you as a researcher want to predict patient survival in hepatocellular carcinoma studies. Most often, and often disproportionately, as both AJ and John talked about earlier, the focus is on the ML code that will be used to solve this problem. And while that is really important, what we often miss in that conversation is that before you can get to the ML code, you first need to navigate this landscape where you have to ask and answer several questions about the data itself. And the eventual success or failure of often the whole project will depend not just on the ML code and the algorithm or the analysis that you're doing, but it will be actually determined by what seem like very simple questions about the data. And it's also these seemingly simple questions that take the most amount of time, effort, and energy to solve. And if you're a data scientist yourself, you would appreciate often are also the most frustrating part of the whole process. They certainly are for me as a bioinformatics scientist. So these questions are, they span a range. They're not all, of course, the same type of questions. You may start, when you start a project like this, you may start with asking which data do I want to include in my analysis? Out of these data that I want to include, are these available to me or not? If they are available to me, where exactly are they available? How can I interact with those data? Are there packages available, or do I need to go to some user interface to be able to download them? Once you figure out which data are available and where they're available, you will then ask the question whether these data contain the outcome that I want to study or not. In this case, that's survival. If it does indeed contain what you want to study, then you want to know how many samples have this data been collected for. Because if you're an ML researcher, you often need data with large number of samples. After that, you will then still need to go inside that data and figure out exactly which columns contain this metadata that I need. Are the metadata associated with the processed or raw data, or do you need to do something to figure out which sample has which survival, and so on. So there are many such questions that need to be navigated before you write that first line of ML code for your project. So let's start with this often the first question in any project, which is which data do I want to include? So I'm looking, I've decided that I'm looking at hepatocellular carcinoma, and because I want to predict survival, I'm looking at hepatocellular carcinoma in Homo sapiens. Now, in this particular case, it may be a seemingly easy problem because most researchers are aware of the Cancer Genome Atlas, which is on the spectrum of ML readiness. It is a fairly well-structured and well-curated resource. So you may start from the Cancer Genome Atlas and you go and look for hepatocellular carcinoma samples, and it's really well tagged. You will be able to identify exactly which patients, how many patients were studied and what data were collected for them. However, if you decide to broaden this search and look for sources beyond the Cancer Genome Atlas, often the search results are so non-specific that they are impossible to navigate. So on the right here, I've shown an example from Gene Expression Omnibus, where if you execute a similar search there, you get 40,178 results. And these include samples, data sets, pretty much everything. But the quantity of it is so huge, and because they're not curated, it's again, I may look at 100, I may even look at 1,000, but it's impossible to navigate all of them. So you can see that metadata standardization makes a huge difference in whether you're able to find what you're looking for or not. In the Cancer Genome Atlas, which is fairly well standardized, you will be able to find what you need. Whereas in something like Gene Expression Omnibus, which is not well standardized, you'll have a lot of trouble discovering what you need unless you already know what you're looking for. So at this point, I want to take you and give you a glimpse of what this kind of search looks like in Liver OmixAtlas, where we have done some of this metadata standardization. So I hope you can see OmixAtlas on my screen. So when you log into Poly, which is our cloud platform where Liver OmixAtlas lives, you will see that we have a total of 389, slightly over 389,000 ML-ready data sets, which are coming from 49 different sources, and they contain 20 different types of data. So out of these, we are only talking about liver related data today. And if I go to Liver OmixAtlas, you will see that I have a total of 6,760 liver data sets, and they're coming from many, many different sources. They're coming from gene expression omnibus, from links, from TCGA, from metabolites, from CCLE, CPTAC, many, many different resources. And they are also distributed across many different types. And this curation also allows us to get an overview of what data from all these different sources contain. So we can see, for example, that there are several normal data sets. There are hepatocellular carcinoma, fatty liver, non-alcoholic fatty liver disease, and so on. And we can also see that most of these data are in humans, a lot are in mice, some are in rat, and then there are other organisms. So from here, I will go to what the data actually look like. So you can see when you open Liver OmixAtlas, you can again see that all of those 6,760 data sets. Each of those data have samples within them which have also been curated. So you can see some of the curations that we have done here on fields like disease, organisms, tissues, cell line, and several others. So now I want to show you what a search like the one that we did on TCGA or on Geo looks like here. So all the data sets from Geo have an identifier that starts with GSE. So if I simply search for GSE, I will be able to see how many data sets from Geo are there. And you can see there are quite a lot. So what I will do is I will go and filter for hepatocellular carcinoma and the studies that have been done on humans. And that already narrows down my collection of data quite a bit. Now from here, I know that I want to use these data for downstream machine learning applications. So at a very minimum, I want at least my data to have at least 100 samples. And when I search based on that criteria, I get about 433, which is now a much more manageable number for me to navigate compared to 43,000 that I had earlier. I also want to point out a couple of other things that curation allows you to do. So if we scroll through and look at some of these data, you will notice that some mention the term hepatocellular carcinoma, others don't. Others use the term, for example, HCC to refer to hepatocellular carcinoma. And that's pretty typical of how you will find most data, whether they're within your organization or if you go to any data repositories out there. And what curation also allows us to do is to not worry about exactly what term somebody may have used to describe a particular disease. Because we have standardized our metadata, if we search for hepatocellular carcinoma, we'll also find cases where the exact term, which in a generic search may not appear. And the other thing, sometimes we also find that people will refer to hepatocellular carcinoma as, for example, malignant liver. And because of curation, even data sets like this where they don't mention any other term except that malignant liver has been used. If hepatocellular carcinoma is mentioned elsewhere in the study or in the metadata, we are able to curate it and identify those samples as well. So with this, I'm going to jump back to my presentation and just kind of summarize what we have done. So what we have done on Liver OmixAtlas is we have currently curated 9 metadata fields. Some of those you saw just now, disease, tissue, cell line, cell types, organisms. We've also curated some very specific fields called like genotypic perturbation, if genes have been knocked out or knocked in. We're also curating what drugs have been used in these data sets, and this list is growing. So what this curation really allows you to do at the end of the day is to discover data sets that are more relevant to you. And it eliminates the non-specificity of repositories like Geo, where you will get a large number of results. So now I'm going to the other component of ML readiness. So if you recall, we talked about two major components of ML readiness. One was the curated metadata, which we have just seen, and the other was the consistent structure. Now, TCGA, as I mentioned earlier, is a really well-structured, well-curated repository. But if you start looking at different types of data within TCGA, you start discovering some heterogeneity even there. So in this example, I'm looking at RNA-seq, miRNA, and methylation data from TCGA. And TCGA provides an R package that you can use to talk to this data from wherever you're working. But if you download RNA-seq data, they will be downloaded as an R class that you need to know what that is. But if you work with miRNA or methylation data, they're downloaded in a different format. And similarly, if you look at the availability of metadata, when you work or import these data from TCGA, for RNA-seq, they're really well structured. The metadata and process data are together and are already combined. But for other types of data, you have to download them separately and then figure out exactly how to link them. I want to make a point here that here we're taking an example of a source that is, as I said earlier, is really well structured already. And if this is the only source you work with and you have figured this out once, this won't be a problem for you. Right? This is fairly simple. There are 3 types of data. There are 2 different formats. You can figure this out. But these inconsistent formats really start to become a problem when you start working with data at a different scale. So either data from different sources or data of very different types. So in Liver OmixAtlas, of those 6,760 data sets that you saw, in those 10 data sources, there were 20 different formats of data that we standardized. And we had to use over 100 different packages to actually work with those formats and data available in those sources. And this is not just about really figuring out a new package or trying to understand a new format. This process of data wrangling is it's really expensive in terms of time, in terms of effort, as well as in terms of money. So to do this, to work with these 20 different formats, we had to use over 100,000 hours of machine time to really standardize all of them and make them available in a consistent format. And later on, again in the demonstration, I'm going to show another demonstration of how this structure can really help you as a researcher work with data from very different sources without really worrying about what is the format of this data and what package can you use to really talk to that repository. So we've looked at both parts of machine learning readiness, which is the curated metadata, and the second part is the consistent structure. And when both of these come together, that's when you get ML ready data. So all data in Liver OmixAtlas looks something like this. So you have curated metadata associated with all of your data, the tags that you saw on Poly. And within each of these data sets, there are samples which have their own metadata that have been curated. And they have molecules, and molecules themselves can also have their own metadata. For example, genes may have different identifiers, they may have different descriptions, and so on. And for each of those molecules, you will have measurements associated with those molecules in all of the samples. And most data on Liver OmixAtlas is structured in this way. And when we do this metadata curation and data structuring, we really focus on we really focus on extensive coverage of metadata. So we started with curating fields that allow you to narrow down and discover what you need. So we started with fields like disease, drugs, tissue, and organisms, and we are expanding that list every day. The second thing we really focus on is depth and specificity of curation. So what that means is we curate our metadata with standardized terms that are consistent irrespective of where the data are coming from. And because we are doing this at scale for, we have already done this for over 380,000 data sets, we need to really make sure that the accuracy of this curation is on par with human experts. And we do that throughout this process of metadata curation. And once that curation is done, we put the process data and curated data together in one place, we link them together, and that's when you see the data in Liver OmixAtlas. So from here, I would now like to show you an example of how you can take these ML-ready data and work with them in your own computational infrastructure. So as I mentioned earlier, many organizations, whether big or small, and even people, even individuals have their own preference of where they want to work. And again, those preferences may be due to convenience, they may be due to privacy, or they may be just due to familiarity, because that's what is widely adopted. So the Liver OmixAtlas that you saw on Poly, all of those data, you don't necessarily have to be on Poly to be able to work with those data. You can take these data and work with them on whatever infrastructure or data science platform you're using internally. So as a proxy of that, I'm just going to go back. Sorry, just trying to get the Zoom bar out of my way. All right. Okay, so as a proxy of that, yes, as a proxy of that, I'm using Amazon SageMaker today, which is a pretty standard machine learning platform. You may have your own data science platform that you have built, or you may be using a commercial solution. What is common in data science platforms or machine learning platforms like this is that they often provide you notebook environments where you can install your packages, write your code, build and train your models, and deploy them. So SageMaker does exactly that. And I've already created a notebook here that takes the data from Poly, and I want to walk you through how we can take these data from Poly, and then how we can use those ML-ready data to answer the example problem that we've been talking about so far, which is predicting survivability of patients with hepatocellular carcinoma. So the end result that we want to get to is the prediction, right? So it's the ML code, but as we saw earlier, we have to navigate all of these questions before we can get to even writing one line of our code. So just a disclaimer before we go through the demonstration, this is not how to do machine learning a demonstration. So I'm going to skip over a lot of parts in this notebook. But if that is something you are interested in and want to understand more about how we build these models, what we are doing, how we're using SageMaker, please drop us a line and we'll be happy to talk about that afterwards. So as you typically would do in environments or platforms like this, you would set up your notebook, you would install whatever you need, you will call whatever functions you need. So here I am working with PolyPython, which is Poly's Python library that allows you to talk to Liver OmixAtlas data on Poly and work with this in your own environment. But the more exciting part is, of course, the data. What does the data look like and how you can work with it? So you can connect from your environment, again, SageMaker in this case, to Poly through your authentication credentials that you use to typically log into Poly. But what is interesting is this part. So the metadata curation that we have done, what that allows you to do, just like the filtering and searching we did on Poly, you can do the same through code here. So as an example, I am searching my curated metadata field called disease for this term hepatocellular carcinoma. And I'm searching for all the data that have been tagged with Homo sapiens. So if I look at the results, there are 1,184 matching data sets, and they come from a variety of different sources, and TCGA will be among them as well. So if you didn't know that you wanted to work with the Cancer Genome Atlas, you can look through these data, filter some more based on what you need, and explore these data more. But today, as an example, we are going to talk about TCGA. And as I mentioned earlier, I'm working with 3 types of data from TCGA: methylation, miRNA, and transcriptomics. So I can now, because I know what I'm looking for, I can straight away go to this field called dataset source and pull out these 3 types of data from TCGA, and I can see all of the metadata associated with them. Now, what code, where code is also really, really powerful and really flexible, is it doesn't just allow you to find which data sets you need, but it also allows you to investigate what samples are contained within those data sets. So what I'm doing next is I am looking inside the miRNA data set from TCGA, and I'm looking at how many samples within this TCGA data set also contain the tag hepatocellular carcinoma, because we know that data from TCGA have disease data, but also associated normal patients. So I can take a particular data set, search for all the samples within it, and also I can ask the question whether the metadata of interest is contained within my data or not. And if it is contained, what does it actually look like? So I ran the search, and I can see that within the miRNA data from TCGA, there are 368 hepatocellular carcinoma samples with several metadata. I've only filtered out a few right now, with several metadata associated with them. And we have the survival column, and the values look like dead or alive. So we, at this stage, we know which data do we want to include. We know how they are available. We know that they contain survival metadata. We also know how many subjects is the data available for. And we also know which columns the survival data is contained in, and what do the values actually look like. So we have, because the metadata have been curated and made available, I've already been able to answer these questions. Now I need to go to the second part and actually look at what is the process data that's available, the data that will actually go in my ML model to do the predictions. So I'm going to skip over the downloading the data part, but we're going to again refer back to structure. So the 2 things that we did was standardize and curate the metadata, and make sure that all data are available in a consistent structure. So any type of data that I download from Liver OmixAtlas will have metadata about samples, it will have metadata about molecules, and it will have the associated measurements. And just as a quick example, I'm going to show you methylation data and what that looks like. So you can see here we have over 396,000 CpG islands, and we have 424 patients here. And if you look at clinical metadata, we will, 420 patients here. And we also have the associated clinical metadata for these 420 patients. And whether I'm looking at methylation data or RNA-seq data, or miRNA data, or even proteomics data, they're all going to look exactly the same. So at this point, we've also answered the question about the format of these data, about the metadata availability, and what the metadata looks like. Now, this may not seem super impressive just for TCGA, just because if you're already familiar with that source, you probably already understand the structure, you probably already understand the metadata. So if you worked with the Cancer Genome Atlas data before, this seems like an overkill, right? You don't need this amount of curation. You can figure out and do the structuring yourself. But as soon as you go beyond TCGA, and we will see an example later, this curation and this structuring starts becoming very, very powerful. So at this point, I've answered most of my questions. I have the data, I have the metadata I need. So I'm going to skip over preparing the data. So you are really the expert of this part. You understand the data best. You can make decisions about what to include, what to exclude. And then once you have decided what to include or exclude from your ML model, you can build your ML model. But the part that is interesting is this. Once you've built your ML model, and you take a result at what the ML model looks like. So here, I'm showing you results for a model to predict the survival of a patient in hepatocellular carcinoma from TCGA. And we're looking at a receiver operating curve here, which means that the true positivity rate is on the y-axis, the false positivity rate is on the x-axis, and the random prediction line is here on the red. And as you can see, it's an okay model. It's not great. And that's okay, right? That's the point. Often the first iteration will look like this. But what ML-ready data allows you to do is allows you to get to this answer fast. And I will reiterate the point that if you're just working with TCGA, this may not be a huge challenge. But as soon as you decide to incorporate some other data type or data source in your analysis, then the first few questions that we asked about the data, they are the ones that tend to consume most time, resources, and money. So as a quick example, if I'm not happy with this model, as I really am not, I will go and look for, let's say, other types of omics that are available to me. And in this case, I'm particularly looking for proteomics. And I see that in addition to TCGA, there is data from one other source, which is another consortium that collects proteomics and genomics data for cancer studies. And I decide to work with proteomics data from this particular source. So I will work with this data in exactly the same way. The data are structured in exactly the same way. So you can see that when I look at the data from this new source, I will have my molecule. In this case, these are protein measurements in rows. I have my samples in columns, and I have the associated abundance. And for each of these patients, I also have all of the metadata. So now I can start to look at the metadata. I can see whether they contain survival or not. And in this case, they do. And so I can repeat the same process. Again, my data preparation is already done for me. I don't need to figure out how to go download CPTAG data. I don't need to figure out what format they make that available in. So I can simply take these data, rerun my model, and take a look at what these predictions look like. In some ways, they may look better or worse. But the point is that you can remove, add new data without really spending all that time in the data preparation process. So this brings us to the end of our session. And I just want to end by summarizing that today we've looked at Liver OmixAtlas, where the main characteristic, the main difference of Liver OmixAtlas is that all the data that are available in Liver OmixAtlas are ML ready. And hopefully now I've explained that what that means is that the metadata and data are structured in a consistent format. and the metadata are deeply curated and harmonized, irrespective of the origin or the source of the data. And you can take these ML-ready data and you can use these in your own computational infrastructure, in your own data science platforms, to work and explore and slice and dice this data in many, many different ways. And with that, I would like to thank you all. And I would like to invite you to come talk to us, explore what data we have that may be relevant to you. Yeah, and Abhishek and John, this is the end of my session. So we can start with the Q&A now.
John, you're on mute.
Yeah, nice job, Shefali. That was great. Let me just change this to gallery view. All right, great. Thanks everyone. That was awesome. And we do have a couple questions. Let's start with the first question here is, most of the sources used in the integration comes with predefined metadata associated with them. How does this integration help anyway? So it's a metadata harmonization question, I think. How is the metadata helping?
Sorry, I'm just reading the question. I'm not sure I completely understand.
So some of the sources come with metadata already associated with it, but it's probably not ideal.
Okay.
How's that metadata helping?
Right. So as I pointed out, that if you are working, let's say, with one particular source where the metadata is already standardized, great, right? But there are very few resources like that. So the metadata may actually be linked to the samples, but that metadata is not well curated. And what that means is, for example, I'll take a simple example. If you're trying to find whether your data contains replicates or not, somebody may use the term rep1, for example, for saying that this is a replicate. Somebody else may actually use a full term called replicate. Somebody else may just say number 1, 2, 3, right? So those metadata may be associated, but in most cases, they're often not well curated. And if you're working with a small amount of data, that's okay. You can figure that out. Curation is not going to be super helpful in that case. But if you are doing machine learning applications where you're working with hundreds of data and thousands of samples, then those little inconsistencies can really make or break your project.
Great answer.
John, if I can add, so we highlighted an example which is on the spectrum of more ML ready, right? TCGA, pretty well studied, very often reused. But the point was that even for such a data set, which is in a very good spot to begin with, there are a lot of painful issues, right? But if you go to something like Geo, you're as well you might as well forget it, right? Because it's just a mess. And I believe, and as we are learning a lot, as we talk to more customers and their proprietary data, they suffer from a similar situation too, where the metadata is not quite associated, is definitely not harmonized, rarely so. And again, that's a challenge that we see is that needs to be addressed to unlock the value of this data.
Right.
So TCGA is pretty good. It's not quite representative of what's out there, right? Even then, it's challenging. That was the point.
Okay. The next question is, are the column values computed by comparing to DMSO/control within the platform, or is that up to the user to create the right subgroups for comparison?
I'm assuming this refers to probably a particular table I showed. It may even be a, I'm sorry, I'm trying to figure out exactly what this is referring to. The comment I will make is that the actual data that you see in the table itself, that will be, in some cases, we've already normalized it. and we have already standardized it. But that standardization looks different for different sources. It's not all done. Like, for example, you have to treat TCGA RNA-seq in slightly different way than some gene expression omnibus data. But we don't really force that on users. So we really specify exactly how we have obtained those data. And if that works for you, great. If that doesn't work for you, you can use the process data in the same structure with whatever processing pipeline you want to use. I hope that's. I am not sure if that answers your question. If whoever asked the question could probably provide more detail, I can give a better answer.
Okay.
Pankaj, John, I can add a little more to this. So, Pankaj, I mean, there's no constraint that the comparisons should only be DMS versus control. So the user gets to decide that. There's another layer which we did not quite highlight is that the processing pipeline where all the RNA-seq datasets, for example, from Geo, should go through the same pipeline, batch corrected, normalized. That's highly configurable. So you may want to plug in your own pipeline, and we can do that. The metadata curation part is what we have, developed our own AI NLP based model to do that. The data processing, the experimental data processing part is highly configurable to your own. And anything that you do on top of that, like in a differential expression analysis, like DMS versus control, that's entirely up to you. Because again, as Shefali said, you would have the most context of what comparisons would make most sense.
Okay, great. Excellent presentation and great job on this accomplishment. What proportion of the data never makes it into a curated data set? What's the casualty rate? Yeah, I'm sure it probably depends on the original quality of whatever data set you're working with, but on average.
So far, I would say we've been able to work with about 90% data that that we have released, but not like, for example, all of those are not standardized the same way. Like in some cases, like that 10%, that 10% is something we are not really able to touch yet. They're fairly complicated to figure out. But of those 90%, we are, in most cases, we're able to pull out this information, but it's just that it's present in very, very different ways. So the amount of time that it, for us to, let's say, out of those 90%, 40% may be relatively simple to figure out. If it's something like TCG, it's very, very quick. But if it's something like gene expression omnibus, it takes a lot of machine hours and
right.
A lot of curators behind the scenes to really do this at scale.
And John, this also is a good moment to talk about, one of the work that Shefali and team are doing is publishing guidelines around quality control, right? What does quality mean? And that says many flavors to it, right? Like metadata quality, the data quality, the model of curation quality, right? And more philosophically, like the approach that we're taking is we are enabling users, just like, you may want to choose what comparisons you want to make, right? You may also want to define quality based on your own metrics. For example, if a given cohort has only one replicate, you may want to filter that out, right?
Right.
So we think our job is to expose that metadata as to how many samples are there per cohort, so that the user can filter on that to define their own, most relevant cohort, right? It's very subjective, and we want to enable that. For some, like 10%, as Shefali pointed out, it's pretty obvious, and we would not bring that to the platform. So that's a waste of everyone's time, right? But for others, like it's more blurry. So our approach is to enable you to make that call with metadata curation.
Okay. No, that's fair. And I, it's interesting. I think there's probably a gradient too of saying, well, yes, we can use some of this data. That doesn't necessarily mean it's going to make its way into predictive models, and it's high enough model quality data. So, but that's interesting. And the question was asked around GWAS data sets are included in the tool, and I think you already answered that, right? You are using GWAS data in some of this. Is that correct? Or
No, currently we do not include
You don't?
GWAS
Okay.
data. That's something that we are working on bringing in.
Yeah,
Okay,
today we
cool.
don't have it, but we will be adding that very shortly for liver and other organs as well.
Okay. The next question is, how would the results for a metadata analysis look like if we want to compare HCC as you showed in the healthy liver, if possible? Is it possible with Elucidata's Liver OmixAtlas to visualize metadata values for normal liver and diseases?
Yes, yes, you can do that. So the liver data that we have, we have HCC samples and we also have normal liver samples. For what I showed today, I had excluded the normal samples from analysis, but that data is there. Those samples are tagged, so you can take them and do a comparison.
Okay, great.
Yeah, for example, like we have GTEx and also some normals within TCGA and GEO, which you can collate together very easily.
Yeah, we
Okay.
also have several single cell liver atlases. We didn't talk about single cell data today, but we also have several single cell data sets from hepatocellular carcinoma as well as other diseases, and just normal cell atlases to explore
Right.
as well.
Okay, great. Another question, how can this atlas help in identifying drug targets? How to identify which databases have the fields of importance? So probably target identification, target validation, that whole space.
John, I would like to respond to this. So we have very real evidence and an example of where a small biotech company in Cambridge has used an atlas like this, not for liver, but for AML, to create their entire target pipeline. Two of them have been validated for two different indications, AML and neuroblastoma. And those validated targets sit on top of a pretty healthy, robust pipeline that is, educating their next experiments and next validation effort, right? So we have one very clean example of where this Omics Atlas for AML has been used to identify and validate drug targets.
Very cool. Awesome. So how would it work where you've got a whole internal data effort? Say you're at a large biopharma, they have their own curated data, you have your product, but now you want to merge that. Just walk us through what might that business model, what that might look like.
Great. No, so John, this is a great question and a very relevant one, right? At the end of this, like, how do you want to bring it to enable our, partners and a lot of folks on this call? So very simply, there are two ends of the spectrum. At one end, if you have a very savvy computational biology team, and we are working with a bunch of such companies, you'd want to take the curated data, public or proprietary. We have done that for companies too, on your own platform of choice, right? So that's one end of the spectrum, which we think is, let's put a label to that, DIY, right? Do it yourself. On the other end of the spectrum, where we, provide our services, our expertise as engineers, as scientists, as bioinformatics scientists, to work with the data that we have structured and curated, right? To find a drug target or find the right patient segment. So this is the spectrum that we have to offer to our customers, and that has served both the parties really well in ensuring that their R&D questions get answered, and companies like small companies like the one that I mentioned on AML hit their inflection points in their R&D journey, right? Because once the target is validated, that's a pretty big deal, right? As most of us appreciate.
Right, right. You would think that some of the .orgs and the public databases and stuff would maybe see some of the things that you're doing and figure out to say, well, why don't you come and help us do this before it gets to the
Yeah.
point where it's becomes a total curation, endeavor,
Yeah.
etc.
No, I think you're right, and I hope it's a question of when, not if, right? And as you pointed out earlier, right, like we are very much in the infancy of all of this, right? So
Yeah,
why curate data post it has been submitted to Geo, right? Why we can intervene much earlier and save a lot of time and money for everyone involved, right? And,
exactly.
that's what we want to do, right? So increasingly we are part of very anecdotal, right? Like some NIH grants where bunch of 10 PIs from different organizations would come together, and the data needs are not quite addressed, right?
Yeah,
And I think we want to have a seat at that table, and we want to help these groups because it helps us. It's very much aligned with our mandate, and it
right.
helps the entire community at large, right? So
Absolutely.
we are seeing this, but I think a lot more movement will happen on this front in months and years to come.
Yep. No, absolutely. And I mentioned it earlier, we're actually looking at data now to add a whole nother dimension onto data to actually dramatically improve the model outcomes and things like that. So that's what's coming. That's going to be that next generation.
Yeah.
But this has been awesome. I think you guys have created an incredible platform. Obviously, there's a lot of really good questions here. We're over time, but that's good. That means people are interested and stuck around. But if there's anything else that you guys want to address, I think we could probably end it.
No, again, I wanted to thank you, John, and all the attendees who showed up and joined and participated. Again, we believe this is the first of many conversations with the committee, so we welcome any follow-ups. If you want to double click or learn more about any specific aspect, we'd be very happy to answer. We have also updated our website to match this webinar. And again, we invite interest, questions, conversations, and we look forward to many more of those in days to come.
Fantastic. Look forward to the next ones. Okay, everybody, enjoy the
Thank
rest of your day and
Thanks.
week. Thanks for everyone's attention and participation. It's been great.
Thank you so much.
All right.
Bye.
Take care.
Bye.
Bye.